In this lecture, we're going to go through several groups: Vibriomonas, Aeromonas, and Plesiomonas. These are somewhat related organisms in the fact that they can cause pretty significant disease. They do share some characteristics with each other. They all grow on MacConkey. They're all oxidase positive, glucose fermenters. But the other thing that they share in common is that none of them belong on or in you. Okay, these are not part of the normal flora. These are environmental organisms, and so they don't belong as part of your flora. So when you find them, you need to report them for the two reasons. One, they're a significant cause of disease, and two, they don't belong there. The family, we're going to go through Vibrio first. Family-Vibrionaceae, genus Vibriomonas. Everybody shortens it, but it is Vibriomonas. This is it's a pretty large group. There are 75 different species known today, and they include again, these are mostly environmental organisms, and you would probably know them better as part of the normal environmental bugs and part of the plankton and of marine environments. Okay, these are symbionts. They live perfectly well with the plankton that inhabits seawater. In particular in warmer climates. They don't do so well in cold environments, but in warmer climates they do fine, and that's their reservoir. When people come in contact with these, they become accidental hosts, okay? The ones, where's my pen? This picture down here is just showing you that the group itself contains most of the bioluminescent bacteria that we know about, and they live as symbionts with lots of those deep sea animals that glow in the dark. The animal doesn't actually glow in the dark, but it harbors a reservoir of these Vibrio, and that's where the glowing comes from. Okay. There's also some rather famous, I guess, toxins produced by this family of organisms, and one of the one that you've probably heard about is tetrodotoxin, or TTX, which is it's a very powerful neurotoxin. It actually is a sodium pump inhibitor, and it's incredibly powerful. You only need a single milligram in order to kill an adult human. But there are several fishes that belong to the order Tetrodontiformes that actually they're the symbiont with this particular group of Vibrio. And so then you've heard of the pufferfish, right? So the pufferfish is a symbiont. It's part of this Tetrodontiformes, and they live symbiotically with Vibrio, and the Vibrio produces the toxin, and the fish builds it up. Okay, so that becomes something of a protective feature for the fish, in this particular case, pufferfish. Okay, so I don't know if anybody here has ever eaten pufferfish. Apparently, it's a big thing in Japan, but I'm not going there. So it's just one example, probably the most famous example of Vibriomonas living symbiotically along with a marine animal. Okay, in this case, the pufferfish. So these two, this toxin that's produced is it's a very well-known toxin, but this is not a toxin that's produced by the human, the guys that cause human infection, which include these basic four: cholera, parahemolyticus, villanificus, and alginolyticus. Okay, I'm sure you've heard of Vibrio cholera before. It does cause cholera, but they all do can cause significant disease, not because of TTX, but because of other characteristics and other toxins that they're capable of producing. So we'll go through those. Now, Vibrio cholera, of course, is the most famous of the human pathogens. It is, again, water bug. These are all water bugs. And although it does prefer marine water, so salt water, it can actually survive in fresh water. So you do have to remember that while brackish or salt water is the key thing, it can survive. So if we were, by some miracle, to have an outbreak in New York Harbor, remember that the Hudson is brackish. So the moving up the Hudson is not impossible for an organism like this. The salt it'll like the salt water, and it can still live in fresh water, so that's not really a problem for this organism. Vibrio cholera is, gosh, the documented outbreaks are numerous. The severe ones have been noted since 1817. We've known about cholera, which is, for a lot of our modern technology. But once we knew about bacteria, then we could trace these outbreaks through, these are things that people kept severe track of. Now, if you look at the countries where we see these outbreaks, you'll note that it's the warmer waters that are at higher risks. Okay, so these outbreaks are a little bit more than just an outbreak. These are actually pandemics that we've kept track of since 1817. So little outbreaks, if it just affects one country, yes, it's noted, but it doesn't make the news like these serious pandemics. So just to give you an example of that, you all remember a couple of years ago we had an Ebola outbreak in Western Africa. Well, what you may not know is that at the same time Western Africa was being absolutely obliterated by a cholera outbreak, which actually killed more people than the Ebola outbreak did. But it didn't make the news because, well, cholera in Africa seemed to have, unfortunately, a very close relationship. One of the things that's associated with these outbreaks, which you might not be old enough, but you might remember Haiti probably the best. They frequently follow major catastrophes, either some kind of environmental or natural disaster that might occur. So in Haiti, pardon me, it was it was a long time ago now. I guess I'm getting old. They had the major earthquake that hit Haiti and just caused devastation. Now, Haiti doesn't have great infrastructure to begin with, which of course is going to put it at higher risk. But following the earthquake, you can see the mess that these people, look at this raw sea and sewage all just swarm together here. Having the combination, we had a significant outbreak in Haiti after the earthquake. Over 200,000 people were affected in that particular outbreak, and that's on top of an earthquake and all the deaths that occurred there. Okay. And then if you go back, you know that Cameroon, all these places that are here, South Sudan, Ghana, Nepal, these are all high-risk areas for two reasons. Lack of infrastructure, meaning they don't necessarily have the greatest public plumbing situation, which would cause the spread from person to person. Okay, so their water supply isn't necessarily completely safe, and neither is their sewage completely contained. And so that's where we get the high risk. And you can have anywhere, just hundreds of deaths, thousands of cases, though, is what we really get in these particular cases. And the biggest thing to note is the warm water is really what they prefer. There have been 7 pandemics, which are, now this, you know, depending on the historian, some might count one as a pandemic where another doesn't. A pandemic is an epidemic which stretches around the world. So it really leaves your continent and it moves to other continents, is when we start thinking of using the word pandemic. Otherwise, it's just an epidemic, okay? And there have been 7 throughout history. You know, the first one was noted in 1816 and it lasted through 1826. Remember, we didn't have antibiotics at this point. But it began in Bengal and it spread across India. One of the things you have to remember is that India at that point was an English colony, and so you had all these British troops moving into and out of India. That helps to move the organism as well. That particular pandemic spread as far as China and Indonesia. And it did actually reach the Caspian Sea in Europe before it receded. And you don't have to know all these different cholera outbreaks. Just know that historically we start early 19th century, early 1800s. And the countries that it affect include pretty much everywhere across Europe and Asia. You have Russia, Hungary, Germany, you know, you have Egypt, UK, you have every country touched by these. And it's through these pandemics where Vibrio cholera gets, like its nickname is King Cholera, because, you know, 20,000 people died just in Paris. Well, I'm sorry, the nickname came from Paris. But you had 20,000 people dying in an epidemic that reached as far as Paris. And the death counts in the various pandemics are, the death count will be dependent upon, again, the infrastructure of the country. It's very difficult to make a count in a country like Indonesia, which is very disparate, and the infrastructure is just not there. To really keep an accurate count. The other thing is that these were all before the WHO, of course. So we didn't have people running to help take care and take care and take good count as well. But you have up to 7 pandemics. The 7th outbreak was actually, I'm sorry, the 7th a pandemic was in early 1900s, 1902 to 1904. Remember, it wasn't too long after that that antibiotics originated, and that actually helped us to control these outbreaks as well. Not to mention the aggressive move to really improve infrastructure across many countries, including countries like India and Indonesia and stuff. The cities do have some infrastructure which helped to control these pandemics. The last outbreak in the United States was in 1910 and it lasted until 1911, and it was actually it originated from a steamship that was bringing unfortunately they were infected, but they were coming from Naples, and it arrived, the steamship's name was Moltke, and it arrived in New York City. But New York City, as you probably know, already had pretty rigorous screening process, pretty rigorous, somewhat my grandmother would say, offensive screening process through Ellis Island, which actually allowed that particular outbreak to be contained. So the people identified the sick passengers on the Moltke, and they quarantined them on Swinburne Island. I couldn't tell you where that is. And so while it did include some Americans, a healthcare worker, for example, on the island at the hospital did die. Only 11 people died in that particular outbreak. Okay, but it is the seventh cholera pandemic, which is probably what we would consider the modern age after the advent of antibiotics. It occurred in 1961, lasted through 1975, and began in Indonesia. And that actually, that particular outbreak, because we had more molecular diagnostics and we had modern chemistry and molecular technology, we were able to look into and study that strain. That strain is called the L-Tor strain. Okay, we actually test now our isolates to see how they're related to the L-Tor strain. Okay, but that it did spread pretty far. It went to North Africa. It hit the Soviet Union, which was around at the time, Pakistan, those kind of countries, and it did actually make it as far as Italy by 1973. Also on the other north sort of eastern direction, it did hit Japan. It did make it pretty far from Indonesia. And there, of course, the information again depends on that we have about deaths and illnesses. Very difficult to quantify if the country doesn't give us the information. The most recent pandemic that we have originated in 1992. It originated in Bangladesh, and that particular strain is the Vibrio cholera strain 0139. So we, at this point, we like to, or we need to, and actually the health department will do it. If you were to isolate a Vibrio cholera, it would have to be sent to the health department, and they would compare it genetically to the L-Tor strain as well as to Vibrio cholera 0139. And the reason is we know that those strains can become pandemics. And so we, because we know that, we need to make sure that we do much more vigorous investigations, much more vigorous quarantine when we have strains that are close to or are either one of these strains. Okay. We do have an increase in cholera in general in the number of cases, although the spread's not crazy. The increase is there, and that's due to obviously increased travel. So not necessarily that a sick person goes anywhere, but I decided to go to, I don't know, Dominican Republic on my vacation. That sounds wonderful. Warm, salt water. Hi, that sounds great. Oh, by the way, I have a wound on my leg, and that wound is a great place for Vibrio to go. Or I really don't, I eat raw or undercooked seafood, which you'll never find me eating, but people do that. I used to work at a raw bar with a raw bar, so people love to eat raw clams, raw oysters is a big thing, or undercooked seafood. So the whole move to say, oh, let me have my tuna raw. This is all stuff, sushi and all that stuff. You got to make sure that it's coming from a reputable place where there's been no blooms or any kind of over growth of the plankton in the area. At the same time, we also have an increase in our immunocompromised patients. That's always going to put you at risk. Okay, that puts you at risk for everything. So we have to include that in our stats here. Just to show you an example of how a cholera pandemic might occur, this is a map from the CDC about a particular cholera epidemic and how it moved worldwide. Okay. One of the things that's to remember, air travel, boat travel, that actually only increases the speed of spread in a pandemic. It's not required for any kind of a pandemic to occur. We had pandemics before we had air travel. And the reason that we do have it is, remember, this is a marine organism. So it can travel by the seas. It can travel just using the currents that exist already in the sea. So it really, air travel, high-speed boat travel, all of that will only increase the speed. It's not going to have a problem making it worldwide as long as it has access to brackish water. Okay, but one of the things you'll note is that we didn't have a lot of problems. There's not a lot of problems with cholera in Canada, not just because they do have um, indoor plumbing, of course, but also because of the cold water. So people who go to vacation in Canada don't come back with Vibrio. Okay, but you can see they tracked this particular outbreak in 3 different waves, and you can see where the travel that it originated in Indonesia, it looks like, or I don't know, it looks like, where is that? Well, Southeast Asia, and then moved across the globe from there. How is it transmitted? Well, during an outbreak, the organism is actually spread fecal-orally. So, and this becomes a major problem in areas of poor sanitation. So the person taking care of the sick person is at highest risk. Unfortunately, the symptoms of cholera lead to a very messy situation, and it can contaminate your hands, which then, I don't know, you put them in your mouth, or you prepare your food, or you don't clean them well enough, and you end up infecting basically yourself by doing this. So making sure that contact precautions are followed with a patient who has this is very important. Now, in between outbreaks, the organism, remember, it's perfectly fine in salt water, brackish or salt water. It has no problem. It lives there all the time, and you only occasionally get these blooms. If the water becomes contaminated with sewage, that just enhances the ability of the organism to grow. Basically, you're providing its the environment that it likes. Okay, and when you collect seafood out of those waters, then you end up with contaminated food source. Okay, so it's not really, well, it is all fecal-oral in the end, but what happens then is roy oysters are the big one, and you can see those here. Roy oysters are the big one that they're most highly associated with, but they're not the only one. So if you know that a particular area has warm salt water, and this is where your oysters or your sushi, you know, whatever fish you're using for sushi, or your clams or whatever it is you're eating came from, then this is something that you have to be concerned about. And it's also one of the reasons that in the United States, sales of raw shellfish are controlled. So in particular, raw clams and raw oysters, if they are served raw, then you actually have to make sure that we keep track of where they're, how they're sold, to who eats them, where they came from. All that stuff is really very closely monitored, and the Department of Health watches that. For if there's a problem in those waters, they'll come back and they'll find out who ate, and they'll go from there. Now, different from somebody like we talked about typhoid, carriers are not a significant source of infection, and that's because basically if you've got Vibrio, you don't become a carrier. It's not your normal flora, right? If you've got Vibrio, you are pretty sick, okay? So you're not a carrier. You don't really get carrier state with Vibrio. Now, the pathogenicity of Vibrio cholera, again, it's the most famous one, and that's because the cholera toxin allows it to be incredibly pathogenic. The cholera toxin is an interesting toxin. It's one of the A and B subunit toxins. Basically, the toxin itself is produced with 2 different subunits that when they come together, and it's a pretty complicated process, how they come together. When they come together, it's basically a cyclic AMP dump, okay, into the gastrointestinal tract, and it causes just massive diarrhea, okay. Essentially, the toxin, it binds to you can see here what happens. I don't want you to memorize everything about this toxin. I do want you to know that it's an A and B subunit. You can see this, the units here are here. You have your A, sorry, I gotta remember to use my pen. Your A and your B subunit are attached to the same toxin. When the toxin is phagocytized into the cell, this is the gastric endothelium, okay? Then it's going to separate. Here's your A subunit, and the B subunit is off as well. And it's the A subunit that actually is the part of the cytosol. It activates the well, it does, you don't really need to know all this, but it does activate the G protein through an ADP ribosylation reaction, blah, blah, blah. But basically, you get continual stimulation of adenylate cyclase producing cyclic AMP. You get this giant dump of cyclic AMP, okay? So what does that do? Well, cyclic AMP is going to trigger the chloride pumps in the membrane. Now, just to give you an idea of what the dump looks like, your cyclic AMP levels inside a cell with the toxin in it increase 100-fold, okay? And so your chloride pumps, you know, you got all this going on, your chloride pumps, all of your chloride is just dumped out into the lumen of the intestine. When the chloride goes out, what follows? Water. Water follows, okay? Water is going to bring with it a whole bunch of other electrolytes, okay? So you say, wow, okay, so water and electrolytes in the lumen. But the amount of water and electrolytes is massive. And so the patient actually, if they die, it's usually from the dehydration. The dehydration is so significant that it leads to heart attack. Okay, so your electrolytes are so screwed up, and you lose everything. There's other virulence factors with Vibrio that can apply not just to cholera but to several others. But the cholera toxin is unique to Vibrio cholera. Okay, I mean, for example, you have just being motile allows an organism to be more pathogenic because it can move along the mucosa, it can find a space to get in and cause problems. Vibrio also produces a mucinase, which actually penetrates the mucus layer. It's not shown in this picture, but there is a mucus layer in the GI tract, and that mucus layer usually prevents bacteria from attaching to the endothelial cells. But with a mucinase, the cholera can make it right through that mucin layer, the mucus layer. And also, Vibrio also produces pili. Pili, remember, those are the structural parts of the bacteria that actually allow it to attach to the mucosal cells. You can't forget, for an organism to produce an infection, it has to attach. That's why just washing your hands doesn't stop, or washing an infection doesn't necessarily stop the infection, because the organism is attached, okay? So just remember cholera toxin and its classification. It's an AB subunit toxin. There's a whole bunch of classes of those. And what does it actually do? It actually causes a massive increase in cyclic AMP, which then drives the chloride pumps, which force all the chloride out of the cell into the lumen, and you get this incredible watery diarrhea, which is characteristic of Vibrio cholera. The disease itself is, as I said, you get watery diarrhea, and what it'll resemble is called rice water stool, which is basically a very light brown colored or tan colored, incredibly watery stool, and the patient will just be losing water incredibly. The stool itself, frequently it might not have any smell because there's so much water in it, but it can harbor a fishy smell, and it may contain some mucus bits. Those are things that are have been reported with cholera. Um, the patient is losing between 10 and 20 liters of not just fluids, but fluids and electrolytes per day. They're usually not going to have a fever. This is a fairly localized infection for the most part. If the patient does have a fever, unfortunately, they probably have a secondary infection on top of it. But because they've lost so much water, so many electrolytes, they're going to be lethargic. And one of the things that you can see, I included this picture, you can see in both of these patients, if you look at their eyes, you can see that their eyes are sunken. And that's very characteristic of cholera, okay, is the sunken eyes. And it's because they're losing water. Their mouth, obviously, they'll have dry mouth because they're dehydrated, cold, clammy skin. Their skin will lose its turgor, you know, that elasticity. Their hands and feet will become wrinkled as if just like you would when you're dehydrated. And then you have this breathing pattern, which is called Kussmaul breathing. This is a very deep and labored breathing pattern. And what's happening is because you've lost so many electrolytes, and in this case, what's causing the Kussmaul breathing is the loss of bicarbonate. And when you lose that much bicarbonate, you're actually your blood is becoming acidic. And so you actually have lactic acidosis with poor perfusion, which is causing these breathing problems. Your blood pressure is going to drop. You're losing volume, okay? Because of the dehydration, your pulse is going to become rapid and thready. You're going to stop producing urine. You're going to get all the other symptoms: muscle cramping, weakness, altered consciousness, seizures, and then coma, which will eventually lead to death. Okay. All of this depends upon, again, the medical infrastructure. Can we treat this patient? Keeping a patient, you'll note that these patients that are lying here, you can see you see this line here on this patient here. You can see the lines. They're trying to keep fluids in these patients, but you can see while the older man has the line, there's no line on the kid yet, and that's because of the lack of resources that this particular area has, okay? You got to keep the fluids in these patients so that they don't die, okay? But the severe fluid loss will lead to what's called the blue death, and that's because the extreme loss of fluid will cause your skin to take on a bluish hue, okay? What are the treatments for cholera? Well, fluid replacement is the major one, okay? But then we can treat to shorten the infection. This would be, in its natural state, this would be basically a self-limiting infection because you're not its normal host. The infection itself will be short. Unfortunately, the clinical situation that you're going through is so severe that you may not survive even a short infection. So treatment with antibiotics like doxycycline or tetracycline will reduce the duration of the disease, but you still are required to make up those fluids, okay? Now, this is the majority of the clinical symptoms and the eventual cause of death are due to the toxin. So maybe you're thinking in your head now, well, why don't we just produce a vaccine to the toxin and then the bacteria really won't cause us any problems? Well, there actually are vaccines available, but unfortunately, they only have a very short-term effect. And they're not widely recommended because of that short-term effect might give you, I guess because it might give you a sense of safety when there is none, okay? The bacteria is able to change the toxin itself, you know, antigenically slightly enough that it can, it will not give you 100% coverage. And again, the short-term effect of the vaccine has not proved to be very useful in prevention. The best prevention is increased infrastructure to prevent contamination of local waters, and then also very good education so that patients know what the levels of the bacteria are in the local environment and that they don't eat raw or undercooked seafood from that area. Now, I'm going to go back again to the strains. We know, again, I talked to you before about these epidemic strains or these pandemic strains. The somatic strains that we're looking at are 01 and 0139. These are the strains that almost always produce the cholera toxin. You can have strains that will produce, some strains of cholera can cause cholera, but they don't necessarily produce large amounts of the toxin. Well, 01 and 0139, they always do. The 01 strain has two separate biotypes, the classic and the L-tor that we talked about before, okay? Any strain that you isolate that is a non-01 or non-0139 strain, they don't produce the toxin, so you're not going to get that same pandemic. You still can get sick, but you're not going to get a pandemic, okay? If they don't produce it at all, you're not going to get cholera because it's really the toxin that causes the problem, okay? And if you have cholera without the toxin, can you still get diarrhea? Can you still get gastroenteritis? Yeah, you can get those, but you won't get that pandemic. You won't get that rice water diarrhea. You won't get that massive dump of chloride and then water and all the other electrolytes. So you're just going to get your normal, everyday, and miserable gastroenteritis. So it's different kind of illness, okay? And I'm sure you either have had or know somebody who's had gastroenteritis. It ain't fun, but it's not cholera, okay? And ironically, it's a non-cholera that could be caused by Vibrio cholera, but it's not one of these epidemic strains, okay? And again, these are somatic antigens that we're looking for. Unfortunately, you know how immunologists are. They only give very unimaginative numbers or letters to these antigens. You know, why not name it something like L-tor? We remember that. But 01, 0139, those are the actual somatic antigens, which means non-flagellar, okay? So it's actually an antigen on the wall itself. Now, cholera is obviously the most famous, but we actually can get other Vibrio infections pretty commonly in patients who work in salt water. And these are patients who either work in the salt water or with the things that come from salt water, like seafood or shellfish, because they could harbor these organisms, okay? So the other organisms that cause that, you have your Vibrio cholera that's non-01, non-0139. They can cause gastroenteritis, we know that. And from gastroenteritis, that can become septicemia or, bacteria in the blood. The cholera has also been found, one of the places that you may not think of, in ear infections. And if you think about it, if you swim in the sea and it has been contaminated with, or it has Vibrio cholera in it, it's hard to say contaminated with, but since that's where it lives. Okay, but if it has Vibrio cholera in it, you could potentially get water in your ear that leads to an ear infection with Vibrio cholera. Non-01, okay? So that's something to remember too. It's an unusual site, but we have found it. Now we go through the other ones. Parahemolyticus, okay? This one is often associated with seafood. This one's probably the one that's most often associated, of the non-cholera ones, associated with seafood. It can cause gastroenteritis, but it'll be self-limiting, like most gastroenteritises are. And that's, if you eat contaminated, I don't know, clams, you'll probably get gastroenteritis. It'll run its course, and you may not even go to the doctor. The other place that we have found it is in wound infections, and this would be for this guy here who's doing his work in the sea, and now he's wearing he's wearing the right boots or pants, whatever those things are. But maybe he has a cut on his hand, and then that ends up getting infected. There have been outbreaks of parahemolyticus, just like cholera. You can get a bloom of this particular organism along the coast, and anybody who's in there has the risk. And so you do get outbreaks. We don't really have pandemics, but you can get outbreaks where 10 or 50 or even 100 people in a particular area end up with a wound infection or gastroenteritis. And these usually are concentrated in the coastal regions during the summer and early fall, where your water temperatures are the highest. This particular bacteria wants warmer water. Okay. Again, the parahemolyticus is the one that's most commonly associated with seafood. And the ones that are most implicated, so that you know what to check off your list, include squid, mackerel, tuna, sardines, crab, shrimp, any of the bivalves, oysters, and clams. Okay, so that's a pretty lengthy list. Okay, so what does that mean? Well, if you cook your food, you will not get gastroenteritis. So if you're having clam chowder, you're fine. If you're having raw clams, make sure you know where it came from. Okay, and that the water there is healthy. If you were to eat parahemolyticus in your food, you're gonna get sick within about 24 hours. Pretty quick incubation period, and you're gonna have, unfortunately, pretty explosive, pretty watery diarrhea. You're gonna have nausea, vomiting, all the cramps, all the fun stuff that goes along with gastroenteritis. But it'll be the worst 3 days of your life. It'll in most cases, will resolve within 72 hours. They have had 1 or 2 cases that have lasted longer. The longest I think they found was 10 days. And this is one of the reasons why we tell immunocompromised individuals, don't eat raw shellfish, don't eat raw seafood, because the risk for you if you're immunocompromised is higher than the rest of the population. Okay. And the other thing is that if you are immunocompromised, the risk of it becoming septicemia is higher. So these are things that we really want to make sure patients follow. Okay. Now, as with anything, gastroenteritis requires fluid replacement anyway. Okay. So that doesn't change for parahemolyticus. It doesn't rise to the level of cholera by any means. Now, Vibrio vulnificus. This is actually most commonly associated with septicemia, more commonly associated with septicemia than parahemolyticus is, in particular in patients with any kind of liver disease, which is interesting. Might be a little clue for you in some of your test questions. Okay, so patients with liver disease, we do need to watch out for vulnificus in particular. And it still is found in wound infections as well. Fishermen, they have sharp hooks that have been in salt water. These are the things that cause wounds that can then may be infected. And as a general group, fishermen are not known to be the most worried about their wounds. So they don't necessarily clean them out quickly, but they should, because this is in the water. Okay, this particular Vibrio is interesting because it's the lactose positive Vibrio. Okay, so that's important. The other ones are negative. Cholera throws us off, but vulnificus will be positive. And you can see here in this chart, it's shown with brackets around it only because it may be delayed. Okay, so you always keep your MAC for a second day just to be sure of it in a case where you're expecting that. Okay. The wounds that we see with vulnificus generally are rapidly expanding. So it starts out as a wound, it leads to this rapidly expanding cellulitis, which then could potentially lead to septicemia. So this particular organism has pretty um, uh, easy time spreading in the human body. Okay. Why is it associated with liver disease? Well, it's thought that it's associated with liver disease because many patients with liver disease have increased serum iron, which is necessary for Vibrio to thrive. And actually, the most virulent strains of Vibrio actually can rip the iron right off of the transferrin in your blood. Okay. If you get a wound infection with Vibrio, they actually, untreated, have a mortality rate of approximately 25%. That's pretty dang high for a wound infection, okay? And that's because it's so capable of spreading and becoming septicemia, okay? If it does manage to make it to septicemia, you end up with a mortality rate that increases to 50%. So these patients are at peril of their life, and the death, unfortunately, is so rapidly expanding. If you're, within 2 days, these patients could be dead. So there is not a known optimal treatment for these, other than recognizing, well, first of all, avoiding them, but recognizing them very early and getting them on antibiotics. And even with that, even with treatment, with septicemia, you've got a 50% mortality rate. So this is important to remember. Then we have Alginolyticus, which Vibrio alginolyticus is again known to cause wound infections, ear, eye, and respiratory infections, as well as bacteremia. Most of the cases that we see are in patients who work at least around seawater. Okay, this is the least pathogenic of the 4 in the group, and it is it's the least pathogenic in the group, but it's still a pathogen. It's not likely to cause death, fairly limited infection, but that doesn't include your immunocompromised patients, who it could become bacteremia, which of course always leads to the possibility of death. The differentiation of these organisms from each other you can see this little chart that I made you so that you can tell the difference. You can see the tests that we're going to use. We want to see the indole, the lactose, the sucrose, and the ornithine to help us differentiate between these organisms. But one of the things that's very helpful is a medium called TCBS. TCBS is a medium, obviously it can grow Vibrio, but it's actually used to help differentiate Vibrio because the different species will grow different colors on TCBS. So you can see if you have a yellow colony, you have to be quite nervous, but you can immediately go to your lactose, or you can look at your indole and your lactose to try and differentiate cholera from alginolyticus. In addition, the clinical symptoms will help differentiate those two as well. If it's green on TCBS, you feel pretty good because you know it's not cholera, but how am I going to differentiate parahemolyticus and villanificus? Your lactose is going to do that for you. Okay, so these are it'll also help differentiate it from cholera and alginolyticus. So these are the tests that you're going to need in order to differentiate them, and you have to know that chart. Now, collection and setup for Vibrio, there's nothing particularly special about the collection of a stool or a wound for the recovery of Vibrio. It doesn't necessarily have particularly difficult, it's not particularly fastidious in that sense. If you're collecting a stool, you're going to put it in a Carrie Blair or some other media that'll keep down the growth of the normal flora. But you can't use, one of the things that has been used in the past to collect stools is glycerol saline, but you actually can't use that for Vibrio, and that's because glycerol is toxic to Vibrio. So anything that has glycerol in it has to be eliminated. from your collection scheme. In the United States, we mostly use collection medias like Carrie Blair, which are fine, and you don't have to worry about that. These organisms in particular, as you know, because it's a marine organism, they're halophilic, they love salt. So you would say, well, my media must have to have salt in it in order to support these. But actually, most media has at least a little bit of salt in it. You only need a little bit to keep these guys going, which is why they can survive in fresh water. And the most media has at least 0.5%, and that's sufficient for them. Now, what's it going to look like on my agar? You're unlikely, because in New York City we don't have a major problem with Vibrio, you're unlikely to have TCBS as part, which stands for thiosulfate, citrate, biosalt, sucrose, agar. But you're unlikely to have that as part of your standard stool workup. But you would have a blood and you would have a McConkey. Well, on the McConkey, it's going to be non-lactose fermenter, so you're going to be suspicious because remember, we work up non-lactose fermenters. So you're going to go back to your blood and you're going to do an oxidase, and that oxidase is going to be positive. It's a Vibrio. Okay, so you're going to look for, you're worried about oxidase positive colonies anyway on the blood. So between those two, you're going to have something to look for in the stools that you work up. If you have a TCBS agar, obviously you're interested in anything that grows on it, and the two colors that you're looking for, you can see green, green here and yellow here. Okay, and those you would have to work up to identify on there. The other thing is that it can grow at lower temperatures than your other pathogens. So room temperature incubation makes sense for Vibrio. So if you do have a TCBS agar, put it at room temp. Okay, you know that one's specific for Vibrio, and I've got it at room temp to verify. So once you have something, you say, oh, I'm suspicious of this particular colony. I think it might be a Vibrio. Well, what are some of the things that you can do? Well, the first thing you should always start out with is gram stain, because Vibrio is a curved gram-negative rod. So you want to see these curved, you see them here. You want to see these on your plate. Now, if every single one of them isn't curved, that's okay. Remember that the organism is, even though it doesn't look like it's three-dimensional, this is actually a three-dimensional slide. So some of them are, oh, sorry, that was in the wrong place. Some of them are actually the, like if you look at this one here, down here on the bottom, you can probably see it looks like a straight rod, but actually these might be the two ends here, and it could be curving away from you perfectly so that you just don't see the curve at this angle, okay? So whether it's curved or it's small straight, you got to work it up. If it's oxidase positive, that's another clue that you're going to be looking at. Normally, you would just take this isolate and you'd run it on your Microscan or your Vitek, whatever you have, whatever commercial kit, even API. Unfortunately, the commercial kits are not very reliable. They don't have very strong libraries when it comes to Vibrio. So there are some other tests that should be done. And one of them is called the string test. In the string test, you put you emulsify the organism in a 0.5% sodium desoxycholate solution, which will lyse the Vibrio cells and release the DNA that was inside the Vibrio cells. And if you put a wooden stick or put a stick inside that solution, here's your solution here. If you put a stick in it, you can see it comes up. The DNA actually is pulled up like a string. And let me get rid of this line so you could see that. You can see the string right here, right there. Okay. This is called the string test. And if it's Vibrio, it will be string test positive. Okay. And then the other thing that we do is a 0-129 disc. This particular disc contains 2,4-diamino-6,7-diisopropyl-pteridine, which is a chemical that allows you to separate Vibrio from any other of the oxidase positive fermenters. And you can see on this this picture here, you have a resistant organism and a susceptible organism. Vibrio cholera, any of the Vibrio choleras, both O1 and non-O1, they will be sensitive. So this is potentially your cholera over here, whereas the other Vibrio are resistant to the 0-129 disc, and as well as other oxidase positive fermenters. They will also be resistant. So it's this guy that you're saying, oh my God, that's the worst arrow ever. It's susceptible to this 0139, so I got to do something about that. Okay. So these are the tests that you're going to be running. Now, unfortunately, they have actually in recent years found that there are some cholera that can be resistant as well. The 0-129 disc ain't the best disc, ain't the best test, but it's one of the things that we have in our arsenal. And just to just to point out something, because it has shown up on other tests, remember we said that most Vibrio require salt for growth, and most media have salt in it. So that definitely supports that growth. One of the other tests that you could do to isolate cholera is Vibrio cholera does not require salt for growth. So if you had a salt-free medium, you could see if Vibrio would grow on that. And if your Vibrio isolate grows on that, it could be Vibrio cholera. There is another Vibrio called Vibrio mimicus, which also does not grow in salt, and that's where it gets its name. It mimics cholera that way. But you should remember that there are two Vibrios that do not require salt for growth, and that is a characteristic that can be used to help identify them. So again, for a larger picture here, what are the things that we can use? Here's your chart. I'm not going to read it to you. You should look, but it basically takes that other chart. It includes the growth in salt and the growth without salt on it, so that you mix all of those things in together, and that helps you to identify the species. Unfortunately, because the commercial platforms aren't really all that great, these are things that you do need to do. So what if your hospital doesn't have TCBS? What if it doesn't have all these things? Well, if you suspect it, you send it to your health department and they'll do it. That's how you would work. Now, to move on to some other organisms that cause equally terrible diseases, we have the family Aeromonadaceae. And Aeromonas is the one within that is our major problem. Okay, there have been 14 species described that can cause human disease. The most important among them include Aeromonas hydrophilia, Aeromonas caviae, and Aeromonas veronii. These are ubiquitous in both fresh and brackish water. They've been found fresh, marine, estuarine, all of it. Okay, they've been isolated from produce. We use water to feed the produce. Isolated from animal meat products. We use water to clean the meat products. This is a little bit different from Vibrio in that it's a straight bacilli. This is not a curved bacilli, but similar to Vibrio, it can grow at a pretty wide temperature range, actually wider than Vibrio. It can grow all the way down to 0 degrees. So you can see it can grow anywhere between 0 and 42 degrees. and it is oxidase positive, like Vibrio. It has been even associated with things like well water. Of course, it's associated with seafood, raw oysters, raw clams. Those you all have to worry about. And the key thing is, it doesn't belong on you. This is not your normal flora. Any Aeromonas is the wrong amount of Aeromonas, okay? The major diseases associated with Aeromonas are both gastroenteritis. We do look for Aeromonas in every stool that we isolate, but also wound infections, both with and without bacteremia associated. Now, gastroenteritis, you get obviously the same way we get most gastroenteritis. You've ingested either contaminated water or food. Whereas the wound infections usually result from exposure to contaminated water. So again, either work or leisure in some kind of water where the organism normally lives. Okay, so that is something. If you do have a wound, you probably shouldn't go in the water. Okay. There are some virulence factors associated with Aeromonas. You have endotoxins, hemolysins, enterotoxins, and Herod factors. But the particular role for each of these, as far as they go with the production of the diseases that we associate with them, those are not clear at this point. Okay. And you can see here we have some pretty nasty gangrene, gas gangrene here, and this is just a cartoon because pictures of necrotizing fasciitis are just nasty. So I just put a cartoon in, which is nasty enough, okay? But they'll be rapidly expanding. You can see the expansion of the is out here with necrosis quickly following. Now, Aeromonas hydrophilia is the one that we isolate most often. It is, once it enters the body of the victim, it can easily gain access to the bloodstream, and especially if it's through a wound, and it'll go to the first available organ, where it'll produce an enterotoxin called aerolysin cytotoxic enterotoxin, and that toxin can cause, or ACT, A-C-T, it can cause a lot of tissue damage. And Aeromonas hydrophilia, if you take it in orally, eat contaminated meat or food or water, it'll cause gastritis, which can include a rice water diarrhea, and it can also cause cellulitis, as up to and including necrotizing fasciitis. As terrible as that is, Aeromonas hydrophilia is actually less of a problem for humans than it is for fish or amphibians. So for people who, farm fish or amphibians, I don't know if they farm amphibians, but, fish farms, Aeromonas is a major problem. So it has economic consequences for certain industries as well. Now, Veronii is more often associated with sepsis, usually in immunocompromised patients with either liver disease or trauma, so similar to Vibrio in that sense. And Aeromonas trata it's actually rarely associated with gastritis, but it has been found occasionally in wound infections. Most cases of gastroenteritis, which is where we look for Aeromonas for the most part, are self-limiting. So the patient is going to become pretty ill, up to and including rice water diarrhea, but it'll be self-limiting, so a few days and then you do recover, okay? Now, how do we identify Aeromonas? Well, again, the place where we're going to look for it on stool culture, we're going to look for those beta hemolytic colonies on a blood plate, and you're going to do an oxidase. If it's oxidase positive, right? So this is positive. Then you have to work it up. You have to figure out, what organism is this? You're going to have to rule out Pseudomonas, but you can easily do that because these organisms are glucose fermenters, okay? Also, all of the significant species are indole positive, okay? Now, this won't just grow on your blood and your McConkey. It can also grow on your CIN plate, which is where you usually look for your Yersinia, okay? And it will mimic Yersinia. It'll have that red bullet, a red bullseye in the center. So you would be working that up anyway to determine what it is. In some cases, some of the libraries, some of the databases on identification platforms are not completely updated, and so this can be confused with Elizabeth Kinga meninges septica, and so those are things that you have to take into account. Make sure that your database is updated. These organisms are resistant to penicillins. There's intrinsic resistance, so you can use that as part of your identification. And it's also usually resistant to most of the cephalosporins as well as erythromycin. So what do we use to treat it? Well, we use ciprofloxacin for the most part in the United States and in Europe, and it still works pretty well, but there have been some resistant cases reported resistant to the Cipro reported in Asia. So that's something that we have to keep an eye on and make sure that we dose our patients properly. So here's your identification chart that you want. You can see that the tests that you're going to run are routine tests that you would do on any gram-negative, really, that you grew. Probably the only one that's unique is the growth in 6% salt, okay? You would just take, and you've seen the salt tube before. We use it for Enterococcus. Well, throw your bug in it and see if Aeromonas will grow in it. It shouldn't, okay, except for hydrophilia, which may be able to grow in that, okay? And you can see the rest of these results. I don't have to read them to you, but you should know the tests and what their results are in order to identify. And then finally, we're just going to go through Plesiomonas. Now, Plesiomonas, as you know, is part of Enterobacteriaceae, okay? But I'm covering it here again because it is oxidase positive. It does ferment glucose. It's a facultative anaerobic gram-negative bacilli, and it's motile. These are things that it shares in common with Vibrio and Aeromonas. So if you have something that looks like, you kind of have to keep it in mind. It might just be a Plesiomonas. Plesiomonas still can cause significant disease, okay? This Plesiomonas is also found, it's found in aquatic environments, also found in the soil, but only for either estuarine and freshwater environments, not usually in straight marine environments, okay? It has been found to cause enteric and extraintestinal infections, but it's probably underreported because it does look like E. coli. Now, if you mistake Plesiomonas for E. coli, then you haven't been doing your oxidase. So you have to make sure that you do your oxidase, okay? There have been some outbreaks associated with seafood, so that's something that needs to be monitored as well. So we can see it in contaminated fish and seafood, because it can be in the bodies of, usually will cause significant problems in cats and in birds. But if people who handle their bodies have been infected by it. This particular organism, Plesiomonas, does share some antigens with Shigella sonnei, and so you have to be careful because it may cross-react with the antiseria that we use to identify Shigella species. But again, it's oxidase positive, so you should have done your oxidase, okay? How do we differentiate it from the other oxidase positive? Well, Aeromonas species is DNase positive, where Plesiomonas is DNase negative, so you could use a DNase test in order to differentiate it. Also, Vibrio and Aeromonas are unable to ferment inositol, whereas Plesiomonas is inositol positive. Okay. And so with the other thing that is, if you have a specimen that's likely Plesiomonas, you can always send it out for PCR. You could always send it out to be identified if your libraries aren't doing it. And so we finish off our the major causes of gastroenteritis and more in humans, and we complete the answer to the question, why doesn't Mrs. Shanley eat sushi?